All right, looks like we've leveled out. Let me go ahead and start the introductions. First of all, I want to thank everyone for attending today's AG2PI workshop. This is the final day of a 4-part set of series on teaching and understanding genome to phenome problems, specifically geared towards non-biologists. Over the previous 6 lectures, you all got a crash course in approximately 4 to 8 years of life science research within this space. And today we have 2 great sessions. The first one is going to be by Dr. Goddard on ethics and social considerations as we think about everything that's happening with with genome to phenome based research. And following this, we're going to have another hour where we'll have a set of short presentations by leaders in the field, just to share with you a little bit about their research and where their current problems are. That one, we want to definitely open it up for everyone to ask whatever questions they want to in the genome to phenome space and leverage the expertise of this panel to explain to you anything that you want to know. And so with that, Ellen, I will hand it over to you to start our presentations today.
Thanks. That's great. I'm happy to be here. I think that this is a slide that may have been, some part of it may have shown up in the original overview of the whole session that Chris provided at the beginning. And it's sort of a summary of the kinds of things that I'm going to talk about. I don't really need to go through it, but if you think in the end I didn't address any of these questions, then feel free to bring them up. I'm an agricultural economist in the University of Alberta, so I'll sometimes talk about Canadian data. I have put in some American data, but I will also say that I have been working on the economic and social implications of the use of genomic technologies, mostly on the livestock side. So more of my examples may be livestock related. But where I wanted to talk today was a little bit about where this big public concern about the use of all kinds of genetic technologies, but originally genetic modification in food actually came from. To be fair, on the crop side, people have been using different techniques to breed new plants that have been pretty fundamental to feeding the world for hundreds of years. And I don't think that the industry was actually prepared for the amount of public opposition to the use of this technology that actually eventually arose pretty well globally, interestingly enough. I remember in one project I was working on, we interviewed people on the street in Edmonton, Alberta, about their concerns about genetic modification in food. And I interviewed people in a tiny isolated tribal village in India on the same project. And the concerns were identical, virtually. So I find that fascinating. One of the things, though, that started a treadmill of public concerns about where food came from and how food was produced that was largely focused on crops that were genetically modified at the time started around the time of the BSE crisis in the UK and Europe. I don't know retroactively whether people can actually understand how big a shock it was to the developed country global food system that was pretty heavily integrated with trade when the UK government finally admitted in 1996 that there was a human health risk associated with BSE in their cow herd. They had been loudly insisting there was no human health risk for quite a while. even to the point of having the then Minister of Agriculture feed his daughter a hamburger on TV in 1990. So they were insistent that everything to do with beef in the UK was perfectly fine, but eventually had to admit that it was causing people to die and changing the regulations. This was a huge shock to the system. And around about the same time, there was starting to be more and more discussion about the use of genetic modification in crops. So I really think that if you unpack these two completely different issues, somehow worry and skepticism about the system being safe played a large role in affecting people's attitude towards GM. And we started to see many articles such as this appearing widely in magazines. That's not the article from a magazine. I collect magazines because I look at food advertising, and I was fascinated by this ad, which appeared way back in 1968 in a major women's magazine in Canada called Chatelaine. Just because you can't read the text, it was fascinating to me that they were actually talking about the technology used to breed bananas at that time. And they were boasting about all the benefits. They're meatier, they're plumper, and they've never been sweeter. The peels are tighter. They have much nicer blooms, nicer sheen, all around better appearance. Now, if we could only get a banana to juice like an orange and keep like a coconut, but still look like a banana. It's amazing to me that if we'd continued with this sort of advertising, in lots of ways, it would have counterbalanced concerns about GM that are appearing in this other picture, but we didn't. And as a result, we started to see this sort of stuff appear regularly. This kind of picture with a needle stuck into a piece of fruit, a tomato originally, many other sorts of food products subsequently, has been really a very pervasive image that a lot of people have top of head. The fact that that has nothing to do with genetic modification eludes people. These other pictures also resonated. We started to see increased use of the word frankenfood, which, by the way, has its own page on Wikipedia. It became such a fundamental way of describing genetically modified foods. And we even started a regular annual day of protest marches globally, in the main aimed at Monsanto, who was largely identified as one of the purveyors of genetically modified food. So all of this sort of mushroomed from the mid-1990s on, really escalated when people started to say, hey, wait a minute, the food system's not safe because of BSE, so maybe it's not safe because of other things. We've had a long history in the use of genetic engineering or biotechnology, or however you want to describe it, on the side of developing pharmaceutical products, such as insulin from the mid-1980s. And many other important drugs, including, I would say nowadays, the vaccine for COVID-19. And people are in general not as questioning about the use of genetic engineering. You have to understand that people are under stress, perhaps when they need to take drugs, and so they don't ask so much about the origins of the drugs, or maybe they only take them when they're very sick and they don't ingest food like we do 5 or 10 times a day, depending on the person. But we've almost had very divergent paths in the concerns about the use of genetic engineering on the drug side versus the use of genetic engineering on the plant side. This author wrote that he thought that part of the issue was that pharmaceutical products were aimed at different things than agricultural products. And interestingly, there's always been a bit of a debate about whether we developed the wrong genetically engineered crops originally that were targeted on things that would benefit farmers, but they wouldn't necessarily benefit consumers. I'm not sure I agree with that, and I'm not sure that my research bears that out, but it still is a very pervasive sort of attitude. Actually, the public in developed countries has pretty positive attitudes towards farmers and is reasonably supportive of the idea of them benefiting in some way. But I think the difference in concerns about the use of genetic engineering in pharmaceutical products versus food products has more to do with individuals, people's perceptions of their health. At some point, if your health is really threatened, then you're not going to care too much about where the drugs come from if they're maintaining your life. On the other hand, the perception is always with food that we have a choice. And increasingly over time, this debate about genetic modification in food has tweaked people's interests to the point that a certain proportion, usually a vocal proportion of the population, will say, I am going to choose not to have that technology. As it turns out, the vast majority in the middle, the vast majority of the population is kind of in the middle and neutral about the technologies. This is an example of American research that came out early in 2020 based on late 2019 results. And the Pew Research people generally have a look at how people's concerns about GM foods are going across time. They've also done some recent work on other uses of technology. But you can see from this chart up near the top that although things are moving around a little bit across time, they're not moving around positively, if you imagine it. So 39% of the population in 2016 thought that genetically modified foods are worse for one's health than foods with no genetically modified ingredients. And that had grown to 51% by 2019. And if you look at the people who are expecting negative effects, they really are very different than the rest of the population in their views. My own research, although I'm not going to bother showing you little charts about very much of it, my own research would say we have an almost flat percentage of the population in Canada since the early 1990s that maintains a concern about genetically modified foods. And so almost identically the same percentage of the population. So obviously it's not the same people if I'm interviewing them in 2019 versus 1992, but the level of concern remains pretty flat, not too dissimilar than some of the differences in some of this Pew research. So why is that? And why does it maintain? And why doesn't our concern go away over time? On the science side, people are always saying, look, particularly in places like Canada and the US, we've been using genetically modified foods for 25 years now, and everybody's perfectly healthy. So where is this concern coming from? And that remains a barrier of some sort for the uses of the technology. This is out of my research, and I just thought I would show you one example. Remember, I work on livestock more than on crops, so we don't know so much about people's attitudes towards the use of genetic technologies in livestock. So I was not looking at genetic modification. I was looking at the use of genomics to selectively breed pigs with higher disease resilience. And you could see that more people thought it was not risky as opposed to people thinking it was highly risky. Interestingly enough, vaccination at the time I did that research, which was 2019, was seen to be very risky for livestock. It turns out nobody knows we vaccinate livestock, so I was a bit surprised by that. Probiotics, though, would be a very much more positive thing, and I don't think Canadians know what prebiotics are, so that's why they're neutral about that. So there's a little bit of positivity here. What I was going to say is, it turns out that that you can unpack very different views of the public about different applications of technology. So, for example, getting rid of disease in livestock that you might ultimately eat is seen as a hugely positive trait, and it's almost positive to the point where you don't care what technology was used to produce that trait in the animals. Although some of my more recent research says a 20-year conventional breeding program would still be preferred by people if you gave them a choice. But genetic modification is still viewed highly negatively. The use of genomics for selective breeding is not considered to be as invasive. So the public is a bit sophisticated. I wanted to remind you that on the agribusiness side of the world, there are lots of technologies that are being developed that are unlikely to ever be really successfully commercialized. This is, you know, sort of an internal document in Cargill where they were trying to talk about the barriers to innovation, particularly in food production and agricultural production in Asia. But look how high they see the risk of public opposition to tampering with food relative to other risks. And the World Economic Forum comes out with a CEO set of risks every year. And up until the pandemic, people's rejection of technologies in food production was seen to be a really significant risk by a majority of the CEOs. And this was not agribusiness firms, this was CEOs of global companies generally. So this concern exists, continues to exist, and can drive investments in new technology. I think sometimes, though, we shoot ourselves in the foot because we contribute to things and participate in things for the best intentions, but we might be sending the wrong message to the public. In the United States, you probably are aware that over the past 10 years, there's been quite a few referendums on whether we should have mandatory GM labeling on all particularly processed food products. And as a result of that, there was lots of money spent fighting referendums in various states within the US before it became a federal mandate. So for example, back in 2015, there were numerous newspaper articles showing how much the big agribusiness firms were willing to invest in stopping people for voting for mandatory GM labeling. Scientists also came out and said they were not in favor of mandatory GM labeling at various times in the past. You have to think about the public who's sitting there saying, I've heard of it, it didn't sound positive, and now there's all these people who don't want me to know about it. That's hardly likely to make the public think this is something that's really safe and should be encouraged. Their reaction to all of this sort of stuff going on is likely to be, why are they hiding something from me? One of the really fascinating stories was, though, when Vermont passed its mandatory GM labeling rule and said that they would implement mandatory GM labeling as long as a couple surrounding states did likewise. Many of the food companies gave up at that point and started voluntarily labeling because they realized they couldn't do separate runs of products just for Vermont. It was too small a market. And it was quite fascinating. So some of these voluntary labels were appearing for almost a year before the mandatory ones were due to be put in place. Not for us in Canada, unfortunately. And what happened is equally fascinating, and I'll show you in a minute. I will tell you, though, that much of the research done on the demand for GM labeling, like this research out of my program that was done way back in 2005, has been done in a context that you can have mandatory GM labeling without any cost associated with it. As soon as you put a cost associated with it, people become very neutral about whether they need to have GM labeling. But what actually happened in the US when we had this voluntary GM labeling being adopted in advance of mandatory labeling being implemented was most of the companies said nothing happened. So it turned out that what people like is the ability to know something. Does not mean at all that they're going to change their purchases, they're going to change the foods they eat. They like the idea of knowing and the ability to find out stuff. And that may be more a reflection of the fact that they were worried that the use of the technology was being hidden from them than that they really hated the technology. So now we get to some of the newer technologies that are being used that are not as controversial as far as the press is concerned, but somehow the way they're being described in the newspapers, I think, is setting the seeds for some kind of potential backlash at some point in the future. Has to be driven by somebody, but so when we describe the use of CRISPR in mushrooms as the gene-edited CRISPR mushroom escapes US regulation, you send the signal that they're getting around the regulatory authorities somehow, which is potentially not a good thing. Nature weighed in 2016 and said the scientists must help to inform regulators wrestling with how to handle the next generation of genetically engineered crops. And I would say we might have a question about whether or not the public needs to be involved in that, informing regulators as well. Collectively with scientists might be a better approach. And even when we see a relatively positive article about Monsanto, who, by the way, the public mostly hated, licensing CRISPR technology to modify crops, right in the headline of the article are the fact that we have to talk about some of the things that need to be restricted because they're potentially negative. So maybe society shoots ourselves in the foot. Maybe the newspapers respond to the fact that they think a concern's there, and that amplifies the concern across time. When we think about the ethical perspectives around genetically modified crops, where most of the literature has focused on, I find it interesting that we focus more on the ethical negative side rather than the ethical positive side. And I'm intrigued by that. I'm not a philosopher, so I don't write too much in the ethical paradigm, but this is the article that I referenced at the top of the page here is pretty typical of most of the articles about the ethics of GM crops. There are concerns about the potential harm to human health, the potential damage to the environment, the negative impact on traditional farming practices, the excessive corporate dominance, and the unnaturalness of the technology. So that sounds like on an ethical basis, we shouldn't be having genetically modified crops. My argument would be, are we actually addressing the ethics that are implied when we don't use GM or other genetic technologies? For example, the global needs for food and for medicines, we are all examples of that today, are driven by many issues. Population growth on the food side, climate change, which is affecting everything, including agricultural production, the development and growth and public health risks associated with antibiotic-resistant bacteria, the plethora of plant and animal diseases that are affecting some of our staple foods. Things like bananas are at risk, oranges are at risk, different types of trees are at risk. Many, many things that are fundamental to what we eat are being affected in ways that could easily be fixed with genetic technologies. If we don't fix them with the use of genetic technologies, is that not an ethical concern when we still have a significant share of the world's population that is consuming nutrient inadequate diets? You can put this all back in the context of golden rice, which was developed and would significantly increase vitamin A intake in rice-eating populations. And it's never been really adopted for commercial use, although many non-rice-eating countries have approved it as a safe food. The major rice-eating countries who still have nutrient inadequate populations have not approved it. And maybe it's a moot point now. We can probably get vitamin A into people other ways. India, for example, has just approved mandatory fortification of vegetable oils with vitamin A to increase the vitamin A intake. But for all the intervening years, maybe so many children wouldn't have had to suffer from blindness associated with vitamin A inadequacies. And I don't think we have enough debate on the ethical positive side of the use of GM technologies, at least in the academic literature. We sometimes see these debates in the mainstream media, but not in the academic literature. Just to remind you about the corporate dominance, this is the sort of picture. There's a wonderful guy at Michigan State University who's been putting together these charts of who owns the seed industry for years and years. And every time he comes out with a new chart, there are fewer and fewer firms controlling more and more of the industry. So this is something that does worry the public, by the way. Fairness and the ability for farmers to be treated fairly and consumers to be treated fairly does underpin a lot of people's concerns once they get past the idea that maybe the genetic technology has a long-term human health implication for them. And the fact that this consolidation is going on is definitely something that we can't deny. Okay, where are we now? Well, certainly these are issues around the use of things like gene editing in babies that we have not resolved to any great extent. We know that even a moratorium on the use of genetic editing for humans for a while didn't work because we ended up with that Chinese doctor going ahead and doing gene editing and all sorts of negative repercussions of that. There are new gene editing approaches to dealing with things like sickle cell anemia that are quite powerful and are being approved by the regulatory system. Is that the same as being used by the public? And are we appropriately handling the public discussions of this? This was something that was recently on the CBC News in Canada. And this American professor was interviewed, and he talked a lot about how gene editing could make social inequalities worse if it was misused. This quote up at the top is quite interesting. The rich could be buying better genes for their children, which would be a nightmare. So this is problematic and needs to be addressed. And we need to understand that when we don't have completely equitable healthcare systems and accesses to the use of technologies, we need to be very careful about the kind of things that are approved. We know in both the US and Canada right now, we're not even having equitable distribution of vaccines in some parts of each of our countries. And surely to goodness, we should, anyway, I'm not going to say any more about that. One other interesting point is that people have written, and this is an interesting article I use in teaching quite a bit, about how the anti-vaccine and the anti-GMO movements are quite heavily linked. And according to this author, they both cause preventable suffering. But what's really unusual is that they both apply the same techniques to get their message across. The anti-vaccine movement, such as it is seriously affecting the number of people that want to get vaccinated against COVID in my province, for example. And it's amazing to me that the pattern is exactly as this author described it about how the information rolls out in my province. And the more we use social media, the less we know about what other people are seeing on social media. And we simply don't know about the various bubbles out there. So I don't think either the anti-technology in food or the anti-vaccine debates are going to be resolved at any time in the near future. But it's worrying that this is continuing to move forward. I wanted to give you one slide about something that has been recently approved on the livestock side. There's been a number of applications on the livestock side, mostly related to pigs, interestingly enough, that have been approved. But this one is really a path breaker. US regulators have okayed these genetically modified pigs to be used for both drugs and for food. And it's interesting that even the people who develop these pigs are not envisioning them being used anytime in the near future for food, although they do think the drugs will probably be developed first. And from sitting around the table with major pig breeding companies on a regular basis, I would say that they're very risk averse about the use of these technologies if they're going anywhere near the food system. And that may be a backlash to something that has happened because genetically modified food has ended up being portrayed in such a negative light, and the concerns about it have been front and center in the media for such a long time. One last thing before I stop, and I'm sure I've talked too long, but we have to think about the role of science in the whole debate about genetic technologies. And I often think about this when I'm training graduate students. We talked to them ad infinitum about the necessity to talk about the policy implications of their research. And if we're doing research on the costs or benefits or anything associated with genetic technologies, the policy recommendations at the end of their thesis always come down to things like, well, we need to educate the consumer and then we won't have any more problems. And we have to say, no, that's not a really useful policy recommendation. And they say, why? But if they just understood, there wouldn't be a problem. That's called a knowledge deficit approach. And essentially, it just literally doesn't work. We have to think of better ways as scientists and as people using the science, who could be drug manufacturers, could be farmers, to talk in a consistent way, in a way that the public resonates with, about the use of the new technologies, publishing more papers, standing up at a public meeting and showing quantitative research results. None of those things are going to be very compelling. We have to talk about the implications for our own lives. This chart, which comes out of a really interesting study associated with a group at Harvard, talks about how when we think that a knowledge deficit approach is going to solve something like vaccine hesitancy what we do is we try and put more scientific information out through press releases, through talking to the media, through getting it into information, and we end up finding out that it actually may polarize people further rather than encourage people to accept the science. So there is a role for science in trying to diminish the effects of some of these concerns, which seem to be permanently lasting. And with that, I will stop. Okay, so that was interesting. And I agree that there were issues before the BSE crisis that had certain elements of the population more concerned than others. Somehow the BSE one affecting meat and killing people was a little bit dominant. In our group, people talked about, in breakout room 2, people talked about some other things. But a lot of it comes down to trust, I think. Does anybody in the group want to add anything other than that? Or do they agree that it's something to do with trust? Trust in science, trust in government that may be driving a lot of these concerns? Or willingness to accept the negative view of the technology, perhaps, is a better way to put it.
Yeah, I would say, because, you know, on the other hand side, I would also say there's been a lot of misinformation that people just they just took it as face value, right? So they didn't understand the science, so they went for the misinformation and followed that.
I was terrified the other day because one of my graduating agriculture students in one of my 4th year classes I teach told me about the study about the rats and the tumors from eating the genetically modified corn. And I'm like, my God, we've been teaching you for 4 years and you haven't filtered that study out of your, anyway, so these things hang around for a long time in people's belief systems, which is quite terrifying. The vaccine people will still tell you it causes autism if you talk to people on the street, and that's really scary. What about question 2, where people were talking about the idea of whether being regulated or not being regulated? Can I just tell you that in the background, there's a lot of concern with how gene-edited animals are going to be treated by the government, particularly in the US. And there's been a lot of pressure to change the way they're going to be treated versus gene-edited crops are being treated. And there have been a lot of newspaper articles where various scientists have been quoted as saying, we don't need the regulation at all. And I worry that that is probably not a message that's going to endear you to the public very well. And I'm not saying, by the way, the scientists actually said that. They're just sort of referenced in that space, and that worries me. So I don't know, should we regulate these things or should we treat them just like conventional breeding and move on? I think it sort of relates to where we are in the debate at the moment. And unfortunately, it's been going on for a long time. So it's different than if it was really a brand new technology.
But, you know, for me, I would say it has to do with the end product of the gene editing. So if the gene editing is used to create a GMO, essentially, so we're introducing, you know, sequences that are not normally in that species, it probably needs to be regulated, just like a normal GMO has to be regulated. But if we're just introducing sequences that are already in that species, or we're just making very small changes that equivalent to mutations and are equivalent to mutation breeding, which is normally has never been regulated, then it shouldn't be regulated. So to me, it depends on, you know, kind of how it's being used also.
I agree. It's very important with the in products. Since genetics products is lots and different compared with breeders been doing for centuries, there's no need to regulate those products, even though the process involving genetic engineering. So it's really up to us to make sure we, guarding our gate and perform everything's right. Regulation is absolutely not necessary for that. Even the GMOs has certain regulations to increase public perceptions and also understanding of ecological impacts. It has its own values, but eventually, public have to understand biotechnology is one of the best breeding methods we can do today, not 20 years ago.
I'm worried a little bit because I'm at least at the fringes inside the science community around this debate. If you approached people on the street and said, hey, technology's moved on and we can do wonderful things with different technologies than what we were using back when GMOs were debated, so we're not going to regulate it anymore. I'm just wondering how that would resonate. I think we have to understand that there's a certain level of confidence in regulation or oversight, even if you don't call it regulation. And if we move too far away from that, then I think suspicion may get generated again. But I don't know. What do the rest of you think?
There's a flip side to that. If something is regulated, it's just like, well, it must be dangerous. Otherwise, why are they regulating it?
I totally agree. It's how you sell the message. I don't know what happens in the US, by the way, but when the Canadian government approves things, there's never a Canadian official running around the country saying they've approved things or boasting about the fact they've approved things or saying how great it is for the country they've approved things. They generally hide the documentation, so you have to click 12 times to find it. So I wonder if the consistent message isn't coming full circle around some of these things. And perhaps when the government chooses not to regulate something, they could sell that message a bit more effectively.
I think maybe the message should be, we're trying to protect you just in case something goes wrong. So kind of not an overlord, a protector. I think that's where a lot of people get twisted, is that when somebody says, I'm gonna regulate this because for whatever reason I don't understand, yada, 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 they assume it's because it's dangerous, but in reality, we're just making sure that, the bridge doesn't collapse. Not all regulations have negative consequences. I think that's just a mental block that people have, and it shouldn't exist.
Some
we
of
have
the
to
debate
understand,
about the vaccine.
yeah. We have to understand that, and I think that's why there seems to be kind of a change in the thought process in Europe around, gene editing as used in plant breeding, for example, that if you regulate a gene edited product, it becomes essentially so expensive to develop because of the cost that are related to the deregulation, that basically no private enterprise could afford, developing and commercializing a product like that, because it would just be, the cost benefit ratio would not be a good one. And so it wouldn't make it. And so it is now realized in Europe, as you see that you can read, for example, statements from the German Green Party or from the French Minister of Agriculture, that they're realizing now that this could become a hindrance to development of new technologies in crop improvement in Europe, and that they're rethinking their policy to be more, as I said earlier, to be more kind of considered as to what kind of product it is really that's being produced by the gene editing, whether or not it requires regulation. So I think at least there's that rethinking process going on because of the cost aspect of the regulation that it will have.
We should talk about the third question, I guess, which was really about how much the public should be involved in setting research priorities or directions, or, I mean, the EU has gone further than most places in terms of now the public is directly a part of risk assessments on the food safety side, which is not very far away from the approval of technology. So I wonder, could we stand a system where the public was involved in helping us set research priorities?
Hey, Ellen, I really like this question, but I am wondering if we hold off on this until the end of the next session, just to make sure that we have time to cover all of our lessons today. And I'm very sorry to cut you off a little short. The other opportunity too is that since we are at the hour, we normally give just a quick 5-minute bio break for folks to get up, stretch their legs. We can continue to discuss this, but I do want to give people an opportunity to walk. And then I'd like to get our next section started really about 5 minutes after the hour. So maybe in about 2 minutes or so. So it'll be a little bit of a shorter break today. But I do also want to just say how amazing your session was, and really appreciate it. And if you can, it would be great to have you hang around for the next 30 minutes or so, which will then let us lead into an open discussion.
That's my plan. Thanks, everybody.
Fantastic. Thank you, Ellen. Everyone, if you'd like just to take a quick break, and then we'll probably get started in a minute or two. And I'm sorry for the short break. And get started on our next session. So for our final session of this workshop series of bringing genotype to phenotype information for non-biologists, we have a series of speakers who are going to be sharing their research and challenges. And the whole idea behind this is for them to get the opportunity to give a quick overview of their work, and to help everyone sort of understand where we are. And if you are interested in these areas or topics or have additional questions, this will be a great opportunity to both follow up on that, and feel free to write down these folks' names. They're all members of the AG2PI community, and actually all part of the leadership team. And the whole purpose of this project, again, is to bridge these gaps between animal and plant and computational sciences, engineering, data science, remote sensing, anything that can fill into this general set of problems. And we want to have an opportunity to be inclusive and engaging with anyone who's interested in contributing. And so our speakers are going to be Jack Deckers from Iowa State University, Jennifer Clark from the University of Nebraska, David Ertel from the Iowa Corn Growers Association, Chris Tuggle from Iowa State University, and Brenda Murdoch from the University of Idaho. And with that, I will turn the presentation over to Jack. Sorry, your title is not up there, but the title of his talk is taking a look at ovine and bovine and genomes.
And I don't know. I don't know anything about that.
I can go. That's my talk.
Brenda, why don't you lead us off?
Oh, happy to lead you off. So, well, thank you for the opportunity to share my research with everybody here. So what I wanted to talk about today is ovine and bovine pangenomes, and really just trying to think about, you know, these two pictures that I have here in front of you are actually have very similar genomes, but I think if you take a look at them, you can see that they're actually very phenotypically different. They've been raised in different environments, and evolution has allowed these two different cattle breeds to be very different. And so the first picture on the left is Highland cattle. It's very adept at and very hardy in cold winter climates. And then the picture on the right is a picture of a Texas Longhorn. He's actually very good at surviving in the heat. And this particular picture that I used is from one of the an animal that has the longest horns on record. But you can see, even just aside from the horns, that their coats are very different. It allows them to do better in different environments. Okay, you can go ahead and click the next. Okay, and then this next picture is actually a Hereford cattle. And so Hereford cow is actually the animal that's used as the reference genome for cattle. This cow, as you can see, actually doesn't have horns. And this cow, what they did way back when is they line bred this animal for a long number of generations in order to get this to be as most homozygous as they could when they wanted to create a very good reference genome at the time. That was the strategy, to make the animal as homozygous as they could to get the best reference genome. Go ahead, Nicole. And so when we think about these three different animals, you know, there's a number of phenotypic traits that we can look at here. But if I just focus on horns for a minute, there's 4 different loci that we know that affect whether an animal is horned or whether an animal is polled. And these loci are not in genes. They're actually outside of genes. They're medium to large duplications or insertions or deletions in the genome that determine whether an animal is going to be horned or whether an animal is polled. And you may have heard as horns as being sort of biological warfare or defense mechanism. And so for that reason, a number of folks have looked at trying to genetically modify cattle to take away the horns or create polled individuals. And in particular, they're doing these in dairy cattle, and that's because dairy cattle have to be handled quite frequently. And we're selling this as a little bit of a safety mechanism. I think you can see that Texas Longhorn, you have a hard time getting in a milking gate, and if you got hooked with one of those horns, it could be dangerous. So when we think about, you know, genetically modifying genes and genomes and genomes to phenomes in particular, a lot of what we know in the genome is outside of genes, and we're just trying to look and harness the energy that harness the phenotypes that have been evolving for many centuries. Go ahead, Nicole. So as I mentioned, Dominette, or that Hereford cow, had been line bred for a long time. That's the way we used to do reference assemblies. But more recently, we've taken a slightly different approach, and that approach is to take very divergent animals and cross them. And so here I'm just showing you an image that I've taken from Corin et al. In 2018, where they first sort of described what we call this trio binning haplotype approach. And so they take these 2 very divergent parental units, they cross them to create an F1, and then that F1 is the animal that they actually sequence. And by sequencing the F1, they can resolve the haplotype of both of those sort of parents. And the way they do this is use short read technology on the parental units and then long read technology on the F1 cross. And then they bin those short reads. You can see in C, I guess I don't have control of the mouse or the pointer. So if you go to C, you can see that the short reads are all binned, and then they get grouped into whatever parental unit, the maternal unit or the paternal unit, and that gets mapped with the long read sequence. And using this technology, they can actually create 2 haploid reference assemblies from that one F1 cross. Go ahead and click. And so this is a lot of the work that I'm describing here is part of the Bovine Pangenome Consortium, and that is being led by Tim Smith and Derek Bickhart, as well as Ben Rosen. And the goals there is to generate high quality reference genomes for all of the bovid species. And, you know, again, to do this, the reason why they're doing this is to look at species specific genetics that might inform us about some biology and some traits that we could use for selection. And in doing this, they're also creating genomic tools. I put Derek Bickhart's GitHub page there. If you want to learn more information about the Bovine Pangenome Consortium, you can go to that GitHub page. And go ahead, Nicole. And so for the sheep pangenome, what we're doing is a very similar thing, and we're taking very divergent sheep and creating these crosses. And so what I'm showing you here is the first cross that we're going to do is a merino. Merino are known for fine wool. So often you'll have socks or mittens or something like that are made out of merino wool. They are semi-arid adapted. And they're going to be crossed with this Awasi fat tail. So these are desert adapted sheep, more found in Asia. And you can see the large amount of fat that accumulates in their tail region or their hind region. That's often used by locals for cooking. Go ahead, Nicole. The next cross that we're doing is East Parisian, which is a dairy breed of sheep. And while most sheep usually have 1 or 2 offspring, some sheep, like this East Parisian, have 4 or 5 offspring at a time. They also breed out of season. And that's going to be crossed with this Navajo churro. And the Navajo churro are often known for their innate horn structure, so they can have up to 4 horns. They can also be pulled, and they're also adapt to arid climates. Go ahead, Nicole. The next cross that we're doing is a St. Croix cross, and they come from the Mediterranean regions and are actually very adapted to tropical environment. They're parasite resistant, and they also have hair instead of wool, these sheep, and that's going to be crossed with a Suffolk. A Suffolk is what we use in the US as a meat terminal breed. You can see that it has some different coat colorings on this meat breed. Next, Nicole. And then finally, we'll do a polypay cross, also a fine wool with multi-offspring that's going to be crossed to a bighorn. So bighorn is a different subspecies. It's Ovis canadensis, and it is actually very susceptible to getting diseases from domestic sheep. They don't have a reference genome, and so we want to be looking at why it's so susceptible to pneumonia. Next. And so that just kind of wraps up a little bit of the crosses that we're going to do. And so this project is really Ovine Pangenome Project is led by a number of the same folks that are in the Cattle Pangenome Project, and we have very similar objectives to deliver high quality genomes for these breeds of sheep so that we can really understand the divergent economical importance of genomic characteristics that result in different phenotypes. And then, so I thought I was going to be following the FANG talk, but a lot of the objective 2 will also lead into better understanding of functional annotation of the animal genomes, which Chris Tuggle will be talking about in a little bit. And then our third objective is to create a graph and a database system so that we can look at these genomes both at a very sort of a larger look so that we can appreciate structural variation, as well as getting right down to the fine tune and looking at single nucleotide polymorphisms, that differences that might exist across these different genomes. And then some of the other pictures that I have on here, like the mouflon, are historic evolutionary contributors to what we now have as the domestic sheep, and also have genomes that we can compare to. And so that's just, we're just trying to understand the power and the information that these various genomes have and how they've evolved over a number of centuries in order to really do better at predicting the physiological outcomes of phenotypic traits that we have in sheep. And so with that, I can take questions or we can move on to the next talk, whatever works.
Hey, Brenda, thank you so much. Let's save questions to the end just to make sure we give all the speakers some time. But if people do have questions, could you please type them in chat? That way Brenda can take a look at them and we can help curate and ask them at the end. And with that, we'll go over to Jack for the next presentation. the title of which is Prediction of Disease Resilience of Pigs Using Multi-omic Data.
Yeah. Okay, thanks, Eric, and good to be with you. Talk about some of the things that we've been working on. And this is actually a project that Ellen has been involved in also, or is involved in, and also projects that preceded this, looking at a specific disease and genomic selection. And so Ellen has been involved in all those projects to provide the GELS components, or the ethical and societal aspects. This looking at disease resistance, resilience, it has implications on for antibiotic use. So if we have pigs that are more resistant or resilient to diseases, the farmers don't have to use as much antibiotics, and that has implications, of course, for society. But also the whole area of use of genomics and animal welfare is all connected to that, and that, of course, has a lot of societal implications that Ellen has been looking at. So this project, funding from Genome Canada and Genome Alberta, and also an industry consortium called PigGen Canada. 7 breeding companies, international breeding companies in pigs, are in that consortium, and they provided the pigs, and they also provided funding. And then we've also been able to get funding from USDA NIFA to do aspects of this project. And so the idea here was to set up a natural disease challenge facility, and it's set up at a research station in Quebec, Canada. And what they do is they bring groups every 3 weeks. There's another group of 60 or 75 young piglets that come from clean farms, so they're healthy. They're entered into this quarantine nursery for 2 weeks, and then they move to a barn that has been seeded with multiple diseases. And so what they did there initially is they brought in pigs from farms where they had health problems, and some specific health problems that are common in the industry. So they brought them into that facility and then mixed them with the incoming pigs, and of course you get transmission. And then the disease challenge is maintained using continuous flow. So normally what farmers do is they do all in, all out. So they a batch of pigs goes into the barn. They grow them to market weight, and then they all are moved out, and the barn is thoroughly clean, so such that you don't get transmission from older animals to younger animals. Well, here, what we did is exactly what we tell everybody not to do, and use continuous flow. So the older batches are still in the barn when the new batch enters this challenge facility, and so you get the transmission and maintenance of this disease challenge. And it's multiple diseases. It's not a specific single disease, but multiple diseases, because that's what happens in the field. There's not just one disease. And the disease pressure here is fairly severe. There is a lot of careful veterinary oversight to make sure that the animals don't suffer unnecessarily, and that it doesn't get out of hand. But it is a fairly severe disease challenge to get maximal expression of disease resilience. And disease resilience is defined as the ability of an animal to maintain performance under disease or under a poor environment. So the idea is that we realize that we can't breed pigs that are completely resistant to every disease that is out there, because most of the resistance genes, there's many genes that are involved in making an animal resistant. And so the approach that we're using is to try to develop pigs or breed pigs that are more resilient. So yes, they may get infected, but they're able to cope with the infection a lot better than other pigs. So that's the idea. Next slide. Okay, so then in the in this natural challenge facility, and we have data now on 3,500 pigs that were sent through this barn. In the natural challenge facility, a lot of detailed phenotypes are collected on every individual. Of course, growth performance, but also feed and water intake. So these pigs are in pens of about 15. They all have an RFID tag, and then there is a feeder that if the pig goes in there, the computer recognizes the RFID tag, and it measures the amount of feed eaten, as well as how long or when the pig got in there and when the pig got out. So every visit is recorded, and the same for water intake. And then we get all the data on mortality, treatments, health treatments that the pigs get, and we take samples from blood and fecal to look at the transcriptome and fecal microbiome. So those are our resilience phenotypes that we're interested in improving. Next slide. But then at the same time, we collect a lot of samples, a lot of information in the quarantine nursery. So when the pigs are still healthy and young, the idea there is that can you measure things on young healthy pigs that predict whether they are going to be resilient or not? Okay, and breeding companies are interested in that because their breeding programs their breeding populations have to be kept under high biosecurity, because if they get a disease into their herd, they won't be able to market germplasm from it. Okay, so breeding companies have a very difficult time collecting data on disease resilience. And so the idea is, well, through this facility, we can collect those phenotypes. And then, of course, with genomics, they're all genotyped for 650,000 SNP genotypes across the genome. We can use genomics to bring that back into the nucleus population that is used for selection. But also, if there are things that they can measure on their selection candidates at a young age in this healthy environment. So is there something about the gene expression profile that we can measure in blood, or the blood proteome, or the blood metabolome, or the fecal microbiome on a young animal when it's still healthy, that is predictive of whether that animal is more or less resilient to diseases? Then that would be very, very useful, because that's what they can select on. Or they can do different immune response assays, either on the live animal, on the animal itself, like a TB test almost, or measuring production of antibodies to a vaccine, for example. Or we take a blood sample and we isolate some of the blood cells, the immune cells, and do in vitro immune assays or phagocytosis tests. There's all kinds of tests that can be done. That can be done on a young, healthy animal without having to sacrifice the animal. Okay, and then of course we want to connect those. So then that's why we have both sides. On every animal, we have these potential biomarkers on the healthy animal and their resilience phenotypes. Next slide. Okay, and so then NIFA, we were able to get a grant to supplement what we do here. And so on almost 1,000 pigs that went through this system, we have 100 immune measures recorded. We have blood transcriptome, so the level of expression of over 14,000 genes. And actually we have that now on 1,700 pigs. 1,700, almost half of the 3,500. We have blood proteome data collected in the quarantine nursery about the level of about 400 proteins in blood, about 40 metabolites, and also the fecal microbiome. And a lot of this, and I should have mentioned this, the lead for a lot of this project is actually at the University of Alberta, Mike Dick and Graham Plastow. My role in a lot of this has been the data analysis and also bringing in some of the additional funding through NIFA. So the question then is, well, how can we best use all of this information to predict resilience? Next slide. So we've been doing a lot of statistical analysis using the 650 case SNP genotypes to see, well, can we do genomic prediction of resilience and then utilize that in breeding populations. So genetic prediction of resilience, of course, looking at trait parameters. So what's the heritability of all these resilient phenotypes? Because it's really a unique data set in terms of its size, and also that it's resilience to multiple diseases that are present. Usually it's there's most of the research is smaller experiments, whereas a specific disease that is that the animal is experimentally infected with. And so here we have natural transmission. So from that standpoint, it's a very unique data set. And then also all the wealth of data we have on these young animals. So the research question is, well, how can we utilize all of this information jointly in the best way to identify which pigs are genetically the best animals in terms of resilience, but also phenotypically. So if we can identify at an early age whether a pig is more or less resilient, then that may determine which barn this animal goes to, for example, or what treatment it gets, whether it gets vaccinated or not vaccinated. So there's, in terms of management, there are also lots of opportunities. So a lot of data, we've done a lot of analysis, but one of the areas we're struggling with is how to combine all this information, all this omics data, to make the best predictions of resilience. And with that, turn it over to the next person.
Thank you, Jack. Our next speaker will be Chris. Chris, would you like to drive this, or are you happy with Nicole driving it?
I think I've got a lot of click-throughs. It probably would be better if I do it. So I think I can try sharing, and it should be set up. So you can see my slides.
We can see your slides. We don't have the voice to text translation, but if you happen to turn that on quickly, great. If not, no worries.
But that's within Zoom.
Don't worry about it. Let's just move forward. Thanks, Chris.
Okay, well, so this talk, as Brenda sort of introduced, I wanted to give an overview of the FANG consortium. And so this won't be specific on any one particular project, but to try to give you a flavor of what people are doing in the FANG project. So it's a global consortium. It started in 2005, and it's grown pretty organically at the level of individual scientists primarily. And currently we have over 470 members just earlier this year. So just a little bit about why I wanted to talk about functional annotation and the purpose of the FANG project. Why is functional annotation important? Well, one of the things that we're trying to do in genome to phenome and to try to understand the real function of our genomes is to look for variants in the genome that might be associated with differences in traits. And so one thing we can ask is trait-associated genetic variation located in or enriched in genes in the gene compartment of our genomes? And that's not too surprising that it is. You can see a positive odds ratio here for GWAS SNPs, or genome-wide association study SNPs, that were significantly enriched for, this is data out of the human field. But what was interesting is they found also a strong enrichment, a highly significant enrichment of these SNPs that are associated with traits in regions that are flanking or outside known coding sequences. So what does that mean? That means we have to study not only the genes, the protein coding parts of genes, but we have to study around those regions and the regions flanking if we really want to understand how to use the variation in our genomes to select and to understand biology. So how do we figure out what parts are important then? So if you think about just a very small cartoon, proteins coding genes are in red, and maybe we would have SNPs associated with muscle growth. And so the idea would be, all right, which one of those is the one we really want to follow? They're all statistically associated. And hey, one of them is in a gene. So maybe that's the right one. But the thing is that gene is not necessarily expressed in muscle. In fact, there may be regulatory regions that turn it on in the mammary gland, for example. Well, so what about regulatory regions in skeletal muscle? Well, so we might have a regulatory region that's where one of these SNPs are, as you can see in the middle there, and that might be controlling a protein coding gene. So that might be the best, but we really want to find all regulation, because maybe there's an unknown regulatory region at a distance from that muscle gene that might be turning it on. So that's one of our, just sort of a cartoon example of what we're really trying to understand. And so this has been worked on for a long time, for over a decade in the human genetics field, genomics field, and we've now adopted that for use by FANG. And so basically the two major things that people look at is what part of the genome is expressed, so where are the RNA levels, and what controls that expression, what pieces of the chromosomes are changed around these genes. Are they more open? Or are they modified in some way? And so this is what they really study. We are studying to try to understand what parts of the region around a gene might be functional. So you can predict the state of the chromatin, the region around it, and you can also create a map of all those functional segments. And so those segments allow us to understand what variation associated with traits is likely to cause that trait variation. So it's a SNP filter for genetic improvement. And so just in a few minutes, I just want to say that the main current activities that we're doing is we're establishing that set of assays on a limited number of individuals. We're developing lots of infrastructure to try to work together and be able to do meta-analyses, including computational tools and bioinformatic infrastructure. And along those lines, as we have resources for this open biology project, we have a FANG website, which lists all of our FANG publications, as well as information on talks like this one that are provided at previous meetings. And we have quite a dedicated data portal, which has a storage of all the information. And this is just an example of some of the early FANG projects that you can see all the species that they're associated with on the left. There's two major pilot projects across species in France and in the University of California, Davis, but there's been many, many projects since then. And this is the data that I don't expect you to read, other than to see that there's about 2,000 experiments in the FANG data portal right now, with over 20,000 different files. All of those are available. In addition, we're slowly putting in about 140,000 data files from legacy projects that were prior to FANG. And that FANG data portal is basically a single access point for all the FANG data sets. And again, it includes that public legacy data. I think the main point I want to make about the resources here in terms of the data portal is that we have very rich phenotypic metadata and protocols that are associated with this actual epigenetics data, so that this really facilitates understanding exactly what kind of sample this came from and exactly how the sample was processed. So if somebody wants to do a meta-analysis of all the different projects, for example, in the pig, or all the different muscle projects across every mammal, then they can do that kind of work, and they can do an appropriate meta-analysis. And along those lines, we're going to have almost 9,000 additional epigenetics data sets that will be submitted in the next 2 years from the currently funded US and European and Australian projects. So we have about $40 million for those projects, primarily from the US and from Europe, but there are Australian and Canadian projects. And we think in the next 5 to 10 years, we'll see an explosion of functional genomics data in cattle, pig, chicken, sheep, and horse, and as well as aquaculture. And what we're hoping to do is move toward a predictive biology to try to increase the usefulness to society and to industry for genomics data. And I just wanted to put together a couple of slides on how you might interact with FANG. We are definitely doing a deep data analysis across species, potentially even across kingdoms, really looking at a deep evolutionary conservation of function. There, of course, there's a very large amount of interest in comparative genomics, even comparative traits across species and looking to see what genes are controlling each of those. Algorithm development, training of students in data science, and of course, the societal impact of using genomics. And so I'll just end there and let you look at this figure. We're out of time, but this is really the future of FANG, the FANG vision for the next 5 to 10 years. All these different projects are parts of those projects are being run by FANG members. And this is really sort of FANG 2.0. And this, if you're interested in more details, we published a paper in Genome Biology last year with sort of setting out our goals. Thanks very much.
Thank you, Chris. Nicole, if you could share the presentation again, just because I'm not sure where the next one is. We will be hearing from Dave Ertl from the Iowa Corn Growers Association talking about the Genome to Fields initiative. And again, I just want to remind people, feel free to ask questions in chat and or think of some to ask at the end.
Okay, well, I'm happy to talk about genomes to field here briefly today. This has everything to do with genome to phenome research, and it was initiated actually back in 2013, which was just 4 years after the maize genome was sequenced. And the purpose was to begin to understand how we can leverage all that genotype information into making improved genotypes and therefore improved phenotypes to go into our farmers' fields, hence the name Genomes to Fields. And so this was initiated in 2014 as a fairly large scale public research program. So next slide. So as you probably know by now, at least, a phenotype is the result of genes interacting with their environments to create unique phenotypes. And so the purpose of this initiative is to combine genotypic information, environmental information, analyzing the genotype by environment interactions to begin to give us enough information to start being able to predict phenotypes from genotypes. And so I'm going to walk through each of these components briefly. So next slide. So we wanted to sample corn genotypes across a wide, very diverse range of corn growing environments. And this map shows you the states where we've had trials located since 2014. I guess this is just a 2020 set, but basically from Cornell University in New York down to Texas A&M and as far north as Minnesota. So obviously a wide range of environments because we want to really test the limits of these genotypes as they're stressed across very extreme environments for their adaptations. Next slide. We've also genotyped several hundred different inbred lines that went into the study. And this scatter plot just shows the genetic diversity in maize. And the gray dots are the existing diversity and the colored dots are those that we have represented in Genomes to Fields. So you can see we've pretty well covered the known, at least the North American maize diversity in this study. So wide range of environments, wide range of genotypes, and then we put them together. Next slide. And then we go out and we phenotype them. Basically, we have a standard set of traits. These are all economic traits that corn breeders collect that are of importance to farmers. You know, agronomic characteristics like standability, flowering dates, morphological features like plant height, as well as, of course, yield. And so we've collected all these traits across all those hybrids in all those locations. Next slide. And then the other, third leg of the stool is the environmental data. Now, in the past, back when I was a corn breeder, we never worried about weather data or environmental data. But obviously, if we're going to understand G by E, we better be measuring E. And so we have weather stations, actually the exact same model of weather station at all these locations. We're collecting data every 30 minutes throughout the growing season. We collect soil samples and measure a number of soil characteristics. And we capture a bunch of metadata, and this may be a little bit underrepresented in people's discussions, but this is very important. It has to do with how the plots were grown, what agronomic conditions were used, and so forth. Just details about how the crop was grown is an important piece of the environment. All right, next slide. This slide shows just a summary of the number of locations and hybrids and unique environments that we've grown in over the last 7 years. So I mentioned this began in 2014, has continued to this day, continuing to collect data. Now, some of the genotypes have changed over the years, but we do have a common set of hybrids in this test that we've grown at every location every year. And so we have this consistent set of genotypes grown across hundreds of different total environments. Next slide. And the objective of this study is to generate all this data and then make it publicly available. So anyone can go out, tap into it, and run analyses to answer questions they have about how do genotypes interact with environmental factors to create phenotypes. And you can go on to our website, which is Genomes to Fields, that's number 2, genomestofield.org, and you can access the data through that. And a number of people have, this is certainly available to anyone who has interest in a large data set containing genotype, environment, and phenotype data. Okay, next slide. So what are some learnings and challenges we've come across over the last 7 years? Well, first of all, we've created what we think is the largest public maize data set ever assembled containing genomic, phenomic, and environmental data. We've also learned that it is possible to coordinate experiments like this across a large number of institutions and collaborators. It does take a lot of work and coordination, of course, but it can be done. To do so, it requires clear protocols, as well as commitments of the cooperators to follow them. And you got to remember that scientists have been trained to think and work independently, so getting them to work together for some of us has been a challenge, but really it's been quite impressive what we've been able to accomplish. Probably the biggest challenge we've run into, and we've seen this in a number of projects, is data volume, veracity, velocity, all needs to be managed. And you got a bunch of plant scientists trying to be data managers. And, so how do you manage data quality control coming from all these different people, measuring data perhaps a little differently? How do you do data cleansing? And then another issue is just transferring large data sets across space, where we want to compile these in a central location. That isn't so bad when you have digital data, but we're starting to collect UAV images where we have terabytes worth of data that we have to transfer. So we have to set up procedures to do that we've never had to before. Then there's finding storage for these large data sets, both temporary while we're still working on cleaning it up, and then long-term storage where we can assign a DOI to it. and then of course, getting it in is one thing, getting it back out is another. And finally, it's the ultimate point is to get to where we're analyzing these data sets. And so people are going to have to come up with new methods of analyzing genomic, phenomic, and environmental data all together. But we've already had a number of successful research projects conducted doing these analyses, and also on our website, you can go and find a listing of all the publications to date that have resulted from Genomes to Field. So I'm going to finish there and invite you to explore the website. Again, it's genomes2fields.org. So I'll let you move on to the next talk.
Thank you, David. Really appreciate that talk. And if there's one, hopefully take home message people are realizing is that we are drowning in data. So there's lots of opportunities, both in terms of management, cleansing, integration, and everything around analysis to eventual predictions. And with that, I'm very happy to introduce our last speaker for this session, which is Jennifer Clark from the University of Nebraska. The title of her talk is Statistical Experimental Design in Plant Genome to Phenome.
Nice. Okay, thank you very much, Eric. So welcome everyone. The place I wanted to start first was to highlight just the area of plant phenotyping in general. So if this is something you're interested in getting involved in, there is a North American plant phenotyping network. It is very interdisciplinary and collaborative. It is free to join. So please go visit the webpage. We have an annual conference that will be coming up next February in person. So you're welcome to attend and learn more about just the area of and science of plant phenotyping. Next. So as a statistician, I wanted to talk a bit about the importance of experimental design of these types of studies. We've heard multiple talks about developing a pangenome, multi-type data in animal sciences. All of these studies involve a tremendous collection of data with some scientific purpose in mind. It may be a group of hypotheses, it may be one specific hypothesis. And so how do we design and operate and sort of enable these studies so that we get the most useful data and we do it as efficiently as possible? So I wanted to go through just a basic example of how this might work. So this is a study of corn genomes in a controlled environment. The idea was that there are many phenotypic traits with underlying genetic and environmental determinants that we know are dynamic. And this is true both across the animal and plant sciences, as well as other life sciences. And so what we want to do is be able to quantify and compare patterns of change in phenotypic traits or genotypic traits in the case of epigenetics as a longitudinal study instead of cross-sectional. So instead of snapshots in time, we look over time and try to understand patterns of change. And so here we're going to look at just one trait of plant growth, which we study as pixel area. So this is plant imaging, where pixel area is kind of proxy for growth. And I'll just use this as an example of how to do one approach to design. Okay, next. Okay, so one way to look at longitudinal data is to take a functional data analysis approach. So functional data analysis is data-driven, non-parametric. Okay, what does that mean? That means we don't make assumptions about the distribution of the data. So we don't assume normality. We don't make certain assumptions about what should the mean expression be. Those things are gathered from the data. So it's very empirically driven. And we want to capture trends. So flexible distributional assumptions, which is good. You can look at correlation over time through a mixed random effects term. You can calculate confidence bands around your estimates, which is very nice. You can look at both treatment. So in this case, it will be well-watered versus drought. And you can look at genotype effects. So in this case, we have 2 different maize genotypes that we'll be looking at. And you can reconstruct growth curves using a relatively small number of measurements over a long period of time. And that's important, especially if data collection is expensive. You want to be able to get as much modeling done as possible, given the amount of data that you can afford to collect. Okay, next. So this is an example of a greenhouse study where we wanted to look at the impacts of treatment and genotype on growth. So RGB imaging, so visual imaging of plants every day. We measure total pixel count, so how many pixels is covered by the plant area. This is 60 plants, so we have 30 per genotype and 30 per treatment. So you can think of it as a cross design, genotype and treatment, 2 of each, 2 genotypes, 2 treatments. We do daily observations of every plant for 20 days. We define day 0 as the day 6 after planting, because day 0 of planting, you really can't see anything yet. So we wait a few days for the plants to actually start growing and then start studying and imaging. So 2 factors, genotype. If you're not familiar with maize, B73 is pretty much the standard. It's been the standard genotype for a long time. And then FFMA is a small, like a mini maize, right? So we're setting this up so that we'll definitely see differences due to genotype and treatment. And we just want to know how well we can capture those. So estimates of treatment effect, genotype effect, and their derivatives. So when you're looking over time and you're interested in change, what do we think of as change for a curve or a line? That's slope or first derivative. So we're looking specifically at the first derivative, right? Rate of change. Okay, next slide. So here's an example, and I realized after I put these pictures up there that I talk over slides, and so I should put more of my talking into text. So if these seem a little bit terse, it's on me. So here's an example of results. So if you look at the left-hand graph, okay, these are growth curves showing well-watered condition. Those are the red curves. We have water stressed or drought condition. Those are the blue curves. And then we have the 2 genotypes represented by the different type of lines. So the solid lines are the B73, the dashed lines are the mini maize. Okay? What we're showing, and you can look on the y-axis, is the first derivative. So this is change in effect over time. Right? So you can see periods of growth where the impact of treatment is going down or going up, or the impact of the genetics is going down or going up over time, right? So clearly the genetic and the treatment effects vary over time in terms of intensity. And on the right-hand side, you're looking at simply the main effects, the difference between the 2 water conditions, the difference between the 2 genetic conditions. So this is really focusing in on what is the impact of genetics, what is the impact of water, right? Okay, so you can see how these effects change over time. Okay, next slide. Okay, this is looking more at what are the possibilities for experimental design. So what we did after we did the study and we collected all this data, we said, what if we had done a more efficient design? And I use efficiency from the perspective of, what if we had collected less data? Would we see a big difference in our results? So scenario 1 was, instead of measuring every plant every day, why don't we measure every plant every other day? And scenario 2 was, instead of measuring every plant every day, what if we only measure half the plants every day, right? So changing the design to be a little more efficient. And you can see from the y-axis where it shows relative difference, that's relative difference in the values of the estimates. That it's really a very small difference, right? For most of the study, there's differences of, 0 to 4% in the values of estimates. So we could have done this study in a more efficient way with very little loss of information. And that's important to show that, yes, thinking about experimental design, particularly in genotype to phenotype studies, where you can control things, can make a difference. Okay, next slide. I'm going to move quickly because I know time is ticking. Okay, one more slide. One more, keep going. We'll just fill up the whole slide. Keep going, keep going. Oh, back one, perfect. Okay, so what are the scientific challenges? Experimental design, optimal design depends on context. And what you've heard today are very different contexts. Every context is different. Every context has different factors, and those need to be considered in picking the correct design for each study. There's observational versus experimental. There's studies where we're simply observing what's happening. And we want to study genome to phenome, but it's not a controlled environment situation. And that brings in a different type of design if it's observational. We have to integrate multiple sources of information. This goes back to Jack's study, where multiple types of omics, and that brings in different analytical methods and techniques. And then there's the dissemination and accessibility of expertise, right? Unfortunately, not everyone who wants to do genome to phenome studies has access to a full-time statistician who can assist with this work. So we do need to put more resources, make more resources available so that scientists can design better and more efficient studies. Okay, last slide. Okay, if you're interested in studying this type of work, I wanted to mention and put a plug in for, there's a graduate program in Nebraska called Complex Biosystems. It has a specialization in systems analysis where we look at exactly these type of data challenges in the life sciences, anywhere from multi-omic studies to proper experimental design, to how to best understand things like pangenomes and phylogenetics, these types of areas. So if this is of interest to you and you're a student or you have students who are looking for areas for grad studies, I encourage you to visit the website and apply. Okay, thank you very much for your time.
Thank you, Jennifer. And all I can say is, please, I hope there's hundreds of people coming out of that program as soon as possible. We've got a lot of work in front of us. So we are at the top of the second hour. For those speakers that can hang around, and if people do have questions, please do. I'll stay online for a bit. And I did see that a lot of questions were being asked and answered in chat. So I don't know if there's anything that's completely outstanding, but if you do have a question, before we ask questions, just a quick round of applause, or thumbs up icons for all of our speakers today. Yeah, does anyone have any questions?
Also, Eric, if people have questions that they think of afterwards, they're free to contact any one of us by email or through the AG2PI webpage and post questions. And we will respond.
Yeah, if you forgot the speaker, feel free to email it out to anyone that you remember, and we're happy to make sure it gets over to the right person.
I actually have a question for any of the speakers here. As genetic modification technology allows us to be able to more efficiently amplify different genes amongst the population, and I'm meaning this especially for livestock, sorry for those of you who are mostly plants, are we going to get an increased issue with loss of genetic diversity, or will it stay the same? I mean, honestly, right now we do have issues with inbreeding that we have to control anyways. Will genetic modification change the game or keep it consistent, that it'll still be an issue?
Well, maybe I can take a stab at this first. I think it's a good question. You know what? You make the genetic modification in one individual, right, or in one embryo, and then you have, just like what you do in plants with integrating that modification. That's what you would have to do in animals also. Yeah, and that would mean that one individual becomes a founder of the population, or a very, very strong founder of the population. So I think maintaining that genetic diversity will become a larger issue. Maintaining that will become a larger issue when we use genetic modifications. Unless you can easily make that same modification in lots of animals or lots of embryos, then you can try to prevent that. But my understanding, as far as the regulations in the US are right now, every new modification, even if it's the same modification but in a different embryo, is treated as a new GMO. So they would have to, at this point, they would have to go through this whole regulatory process for every animal or every embryo that they modify. So they would definitely then want to limit how many embryos they modify, and therefore get into the situation that genetic diversity might become an issue. So definitely.
I have just a couple things to add. So each livestock species industry is a little bit different. So dairy already has a fairly small effective population size, but this is a valid concern, and they do have germplasm banks in a number of different ways that people are trying to make sure that genetic diversity resources are maintained, even if certain lines of pigs or certain breeds of or certain lines of cattle are perpetuated on a slightly larger volume. But they do have germline repositories and a number of different things in order to help ensure that we maintain the genetic diversity that there is out there now. So great question.
Thank you. I also think that there's a lot of potential with utilizing genetic modification to transpose different alleles and maintain them even better in other populations. But I don't know if everybody thinks about that necessarily.
Yeah, so you're saying, well, you can use genetic modification to create genetic diversity. Is that what you're getting at? Okay.
Yes,
yeah,
or like anything we have in the gene bank, we don't always want to utilize heritage breeds or whatever because they're not as high performing as our Holstein breeds, our synthetics that we're developing. But if we want to maintain a certain allele for diversity, we can transpose that single allele over to our higher producers without having to sacrifice any of the other traits necessarily.
Yeah, our biggest challenge is that we don't know enough about the genome, and that's where FANG and pangenomes and so on come in. We want to really get into genome editing and changing specific loci. We need to know a lot more about what these genes do and how they play a role.
Right, and as an example, the horn locust is not actually in a gene, right? So, but there's obviously regulatory elements that are underlying those duplicated regions of the genome.
Well, thank you. That really sums up what I was wondering about.
Excellent. Well, with a pause in the questions, again, I do want to thank all the speakers from today. I also want to thank Nicole and Ryan specifically for really writing herd in terms of organizing this entire workshop series. And I think there will be a follow-up survey that's going to get sent out just in terms of getting your thoughts and opinions. We will probably be running a similar workshop a year from now, and we'd love your feedback to know what it was that worked and didn't work and how we can make this better to help continue to engage a broader community of interested people to help participate with the genome to phenome problem. And with that, I really want to thank all of you for your attention and holding out till well beyond our 2 hours. And thanks for joining us for this entire workshop.